For over 30 years, the Robert Debré Hospital in Paris has been treating neurodevelopmental disorders in children. Dyslexia, hyperactivity, autism, or rare diseases are disorders that are now part of many families' daily lives.
Studies have not yet established the prevalence of these disorders, somewhere between 5 and 10% of the general population of children. People are becoming increasingly aware of what life is like for these children and their families, as well as of the care, support, and rehabilitation that are necessary and urgently needed.
In order to advance research, Professor Sandrine Pasmard raises parents' awareness of the innovative projects in which she participates. The skin donations of her young patients are precious for researchers. She works in particular with this center, specialized in epigenetics and cell cycle. Here, the formation of the brain before birth is being studied to identify a protein that may be involved in a fairly broad group of neurodevelopmental disorders.
The development of the brain is very sensitive, both to mutations in the genes and aggressions that occur in the womb, which are sometimes due to exposure to chemical substances, such as alcohol, or bacterial or viral infections. The brain, especially before birth, is a totally inaccessible organ. No samples can be taken, and until now, not much was known about it.
In recent years, a revolutionary technology has made its way into laboratories, the organoid.
An organoid is a kind of replica of a human organ on a miniature scale that we grow in the laboratory. At the moment, the focus is on skin and liver, but here, we're particularly interested in cerebral organoids, which mimic the brain.
To make these organoids, scientists use modified cells that have been given amazing properties. Harvested from the skin of patients, they have been reprogrammed to become pluripotent, and therefore reproduce the development of any organ. In a sterile room, a new culture of organoids has begun to form on plates made up of small containers called wells.
On this rather special plate, we have placed 9,000 cells per well in a cocktail of nutrients at 37 degrees for 5 days and CO2 at 5%. At this stage, what is important is that they are spherical to ensure that the distribution is even when they start to grow.
The cells are then transferred to another more potent liquid to begin their transformation into brain cells.
On this plate, we have organoids that are larger. They are 40 days old and are dense enough to be observed directly with the naked eye.
From these images, the advantage of organoids is obvious. This is the evolution of cells in a traditional two-dimensional culture called flat culture. The organoid allows the cells to arrange themselves to create a structure.
With a 2D culture, I would have cells scattered across my field of vision that don't organize, don't structure themselves. We no longer have access to that organization of the tissue like we do with the organ.
The team has already generated several hundred organoids in compliance with the ethical rules related to the use of biological material of human origin. Though they reproduce certain properties of developing brains, they are simplified copies, without connection or consciousness.
For example, there are no blood vessels, no blood, so there are no nutrients, no oxygenation, and most importantly, there are no sensory signals. This brain is not connected to anything. It is not connected to sensory organs, hearing, touch, muscle contraction. We are growing something with neuronal activity, but we can't say we are growing a real brain.
After a month and a half, the organoids are cut and placed on slides, which the researchers take to the microscopy room under close supervision. The goal of this immunofluorescence session is to map the organoid to distinguish its components and perhaps identify the protein responsible for certain neurodevelopmental diseases.
Down there, you can see the neurons appearing. What we're doing is marking the organoid with different colors, since it's naturally colorless. We are basically creating the ID of the cells present in this organoid. With this device, we can do a close-up to enlarge the images and compare the morphology of patient organoids to healthy donor organoids.
These analyses may enable a better understanding of cell reactions and dysfunctions that contribute to neurodevelopmental conditions, so that new therapeutic treatments can eventually be developed.
We can really use these organoids to test certain types of molecules. It's possible that they could also facilitate earlier detection and diagnosis. The adventure is only just beginning.
Other research involving brain organoids is underway around the world on diseases such as multiple sclerosis, Alzheimer's, and Parkinson's. In the healthcare field, these mini alleys may very well enable giant leaps forward.